So I'm going to be discussing positive and negative selection of thymocytes. So to begin with, it is double positive thymocytes, CD4+, CD8+, that are going to be the cells that are the primary target of thymic selection. What exactly is thymic selection? What's it doing? Well, basically it's shaping that T cell receptor repertoire of the double positive thymocytes, all based on the affinity of those T cell receptors, TCRs, for the MCH peptides that they're encountering as they're browsing through the thymic cortex. And just a little background information before I go into exactly what is positive selection versus negative selection. Well, in our thymocytes are coming into close contact with thymic epithelial cells, and these epithelial cells express high levels of the class 1 and class 2 MHC molecules on their surface. And these self-MHC molecules actually will present self-peptides. And the double positive thymocytes will actually undergo positive and negative selection, all based on and depending on the signals that they're receiving from when they encounter these self-MHC, self-peptide combinations with their receptors. So now, positive selection versus negative selection. Positive selection is selecting for those thymocytes that are bearing receptors capable of binding self-MHC molecules, thus resulting in something known as MHC restriction. And negative selection is actually selecting against thymocytes that bear high affinity for those self-MHC peptide complexes, and that's going to result in something known as self-tolerance. Well, I have a figure I want to show you to kind of explain. So we're going to start out with the double positive thymocyte up here. And from here, it can basically be considered to go to one of three fates. And it's kind of all depending on the affinity of their T cell receptors for the MHC self-peptide combination that those receptors encounter. So if the newly generated TCR does not bind, has no affinity for any of those MHC self-peptides they encounter, then it will die by neglect. If they bind too strongly to the MHC self-peptide complex, then it will be negatively selected. But if these double positive lymphocytes bind with low affinity, just the right affinity to the MHC self-peptide complex, then they're actually going to be positively selected. And from here, they can actually mature on to the single positive CD4+ or CD8+ T lymphocyte. And so now something very interesting is that the vast majority, about 98% of these double positive thymocytes are never even going to meet that selection criteria, and they're going to die via apoptosis, cell death. So main point to take away is that very small percentage, actually only about 2 to 5% of the double positive thymocytes are actually exiting the thymus as mature T cells. Vast majority of them aren't even making it to the selection. They're going to die by neglect. So what are the cells that are actually playing key roles in these processes? Well, thymic stromal cells. Those include epithelial cells, macrophages, and dendritic cells. These young double positive thymocytes are going to come into close contact with these stromal cell types, and they're basically going to browse the MHC self-peptide complexes that are displayed on these cell type surfaces. And as the thymocytes are migrating through the thymus, they're going to encounter many different stromal cell surfaces, and then they're going to have the opportunity to bind many different MHC peptide combinations. So now I just want to point out a few different highlights about positive selection and negative selection. Positive selection is a process that can actually induce both survival and differentiation of those double positive thymocytes. Excuse me. Something interesting is that the TCR-MHC self-peptide interactions in positive selection are actually at least 3 times less in intensity than those interactions experienced by negative selection, which is very interesting. And it's actually not fully understood the signal cascade events in positive selection of how they're going from those positive selecting signals to maturing on to that single positive stage. But they've thought that positive selection has some remarkable adaptive value. It's been suggested that positive selection increases the efficiency of negative selection as well as the efficiency of peripheral T cell responses. Basically, without positive selection, the system would just be really cluttered with lots of cells that would be unlikely to recognize anything. And without positive selection, it would greatly reduce the probability that a T cell would find its antigen in any amount of reasonable time. So negative selection, what's going on there? Well, you're going to have autoreactive double positive thymocytes that are going to have high affinity receptors for self-MHC, self-peptide combinations. And these autoreactive cells are going to be very potentially dangerous to an organism, and they are going to be killed and eliminated by negative selection. So it's a very important process. Negative selection is simply any process that will rid a repertoire of autoreactive clones, and it's responsible for self-tolerance. And it's thought that the most likely process by which negative selection happens is known as clonal deletion. And what's happening in that is that there's basically just a high affinity for a TCR interaction that wants to directly induce apoptotic signals, cell death. Another interesting thing I found is that actually the clonal deletion of double positive thymocytes has the same, or is mediated by the same cell types and the same interactions as those that activate mature T cells. So what's been a very active area of investigation is why these strong TCR signals are resulting in death of an immature T cell, but proliferation and differentiation of a mature T cell. Very, very peculiar thing. Another thing to point about negative selection is that it has the potential to happen in both the cortex and the medulla. And so now I want to go back and hit on the errors in the negative selection process and what that leads to. So when there's errors in the negative selection process and those autoreactive cells can't be eliminated, then it's actually that's actually responsible for many different autoimmune disorders. And one of those we hear about all the time, and that is type 1 diabetes. So I kind of researched that a little bit more and wanted to talk about that. Well, type 1 diabetes is an autoimmune disorder and disease. And what we know about autoimmune diseases is that the primary cause is actually the escape of those self-reactive lymphocytes from negative selection. And surprisingly, they really don't know much about why these autoreactive lymphocytes have the ability to escape negative selection, even though this is a problem in many well-known diseases, type 1 diabetes, MS, rheumatoid arthritis, many different things. But, excuse me, they've done some research with a mouse strain known as the non-obese diabetic mouse, which they believe to be susceptible to type 1 diabetes, and they've learned some things from this study. Well, start out with type 1 diabetes is a T cell mediated autoimmune disease, and it's caused by T cells that will kill insulin producing beta cell islets located in the pancreas. And the investigators that did the research with these mice actually found out that endogenously produced MHC insulin peptide combinations and the exogenously formed MHC insulin peptide complexes differ in conformation. Because of that, they're activating different T cell receptors specifically. And even more important, these observations suggested a reason for how the autoreactive cells are escaping the thymus without undergoing negative selection. And so what they found is that dendritic cells in the pancreas, but not in any other tissues, appear to be unique in the fact that they're capable of forming MHC class II insulin peptide from exogenous sources. And they think that's mainly due because they are in an environment very rich with that extracellular insulin that's being secreted by those beta islet cells. However, not in the pancreas, thymic medullary epithelial cells or dendritic cells don't have the same capacity and in that same environment. And so therefore, that's why they're sneaking through that negative selection process in the thymus, and why those autoreactive cells are leading to these autoimmune disorders. And so most current therapies for autoimmune disease are focusing on that inhibiting that secondary but very proximal cause of the autoimmune diseases, and that's simply the peripheral activation of T and B cells that escaped negative selection. But they're trying to get down to the primary source and defining the molecular reasons for why these self-reactive lymphocytes have the ability to escape negative selection. And hopefully they're going to find a way soon of how to develop some therapeutic approaches to defects in this negative selection process. And so now going back to the figure to kind of help you understand, well, I had a question of, do positive and negative selection occur at the same time, or is it a sequence? So it's very clear that thymocytes are undergoing positive selection in the cortex, and the medulla is actually the site of negative selection to these tissue-specific antigens. And this compartmentalization of function is kind of suggesting that some thymocytes are positively selected and will initiate a maturation program prior to negative selection. As we can see, positive selected maturing on down to the single positive thymocytes before they enter that negative selection process. And just the fact that this can happen is supported by observations that showed semi-mature thymocytes. And what I mean by that is that those that have been already positively selected and are in transition from the double positive stage to the single positive stage, these semi-mature thymocytes are actually excellent targets for negative selection. And so the most likely explanation for all this is that thymocytes can be negatively selected at more than one point of development. So we know that those thymocytes that bind with high affinity are negatively selected, and that those that receive the positive signals actually are given the permission to mature. But what else they're given permission to is to migrate, and they can migrate to the medulla. And it's here in the medulla that these semi-mature thymocytes can again be subject to negative selection if they interact with a high affinity to tissue-specific antigens. And so what has been found recently is that migration of these positively selected thymocytes onto the medulla is now known to be dependent on expression of a chemokine receptor known as CCR7, and these CCR deficient thymocytes that fail to enter on into the medulla interestingly still mature and they're exported to the periphery, but here they can cause autoimmunity. And that's just another observation that further highlights the very importance of the medulla and removing those autoreactive cells via negative selection. And further on back to here, those double positive thymocytes who were selected to were positively selected and then allowed to mature on to single positive and then allowed to migrate to the medulla. Well, once they have migrated to the medulla, it was here that they were exposed to AIR, medullary thymic epithelial cells, also known as MTECs. And these express tissue-specific antigens and can mediate negative selection. And then medullary dendritic cells can actually acquire MTEC antigens by engulfing MTECs and mediate negative selection. So there you can kind of see where you're going from positive selection on down to being allowed to mature, being allowed to migrate, and that migration to the medulla being allowed to undergo negative selection. And so to kind of wrap it all up, basically thymic selection is involving many interactions of double positive and single positive thymocytes with both the cortical and the medullary thymic stromal cells, as we saw the cortex and the medulla being both involved, as well as dendritic cells and macrophages were involved. And the selection results in mature T cell populations, and these mature T cell populations are going to be both self-MHC restricted and self-tolerant. And feel free to use my video, and that's all.